All right, guys, lab number 9, form and function. So once again, we're talking about monocots and dicots, two major groups. There is more in the flowering plants. However, today on this level of botany, we'll talk about the monocots and dicots generally. So again, we'll discuss the difference between monocots, the words derived from number of cotyledons, 2 cotyledons dicot, 1 cotyledon monocot, and then we are looking at the parallel venation versus the pinnate/palmate venation for the dicots. The vascular bundles will be scattered. You will clearly see it in today's lab, or they will be arranged in the bundle, kind of in a ring, and the multiples in the flowers. So let's kind of go 3 major innovations of the angiosperms, right? Well, what would be the 3 major innovations of the angiosperms? Flowers, fruits, right? And also the food source, the endosperm for the embryo. So flowers to attract the pollinators, fruits to protect the seed, and then within the seed we have the extra food for the embryo, stored food for the embryo, so embryo can develop relatively fast. Do angiosperms exhibit alternation of generations? All plants do. So yes. Is the dominant plant body, let me increase it a little bit. Yeah, here we go, a little bit better. Is the dominant plant body of angiosperms sporophyte or gametophyte? It's not a moss, it's not a liverwort, so it's a sporophyte. The sporophyte. What are the characteristics you will find in angiosperms but not gymnosperms? 2 major ones: flowers, fruits. The seed is enclosed in a fruit, hence the hence the name, closed seeded plants. What are 2 main groups of the angiosperms? And that would be monocots and dicots. Name 3 types tissues in the plants. We just discussed that last time, but here we have dermal tissue, vascular tissue, and the ground tissue. And number 8 is essentially repeating number 5. Oh, number 7, what is the function of vascular tissue? Vascular tissue is the highways, transporting things. Transport part. So transport what? Water and minerals to the site of the photosynthesis, sugar, dissolved sugar throughout the body of the plant. And number 8 is essentially covering the whole entire, repeating the number 5. So 2 groups of angiosperms, monocots and dicots. There is more, but we are focusing our attention on that. So now let's take a look at the model, and you will need to label. So here we're looking at the tip of the root. Each tip of the root will have essentially 4 major parts. First of all, we're looking at the root cap. The cells are either dead or dying out, and the function is to protect the meristem, the embryonic cells, which are dividing really, really fast from what? From any kind of things you can find in the soil, the rocks, the grain of sand, or anything like that. So protection, protecting the cell division zone. So root cap, now we have a cell division zone right here. Then as soon as the cells divide, they start increasing in sizes. So elongation, zone of elongation, division zone, elongation zone, and then they become mature. They start specializing into different types of the tissues, and then quite often you start seeing the root hairs coming out, and that's known as the zone of maturation. What is the function of the root? Protection of the apical meristem or root meristem. Where do you find the root hairs or cells in the apical meristem divided by a mitotic division? Do you think that we would find in the apical meristem? Where? At the very tip of the living root, so to speak, because those cells are kind of dying off. Where do you find the root hairs? The root hairs will be found in the zone of maturation. All right. So now, whenever you're looking at the cross section of the model from the top, so and again here it shows you root cap, zone of division, zone of elongation, zone of maturation. From the top, whenever you're looking from the top, you can clearly see all of those three types of the tissues. The outer layer is the dermal tissue. The central part, especially right here, is the conductive vascular tissue. Everything in between known as the ground tissue. What is the function of the xylem? Takes water and the minerals throughout the body, usually the movement upward, from the roots, from the ground, all the way to the leaves to do the photosynthesis. Do you see all the three types? Yes, you'll see all three types. That particular part is the vascular tissue, ground tissue, and the dermal tissue. Explain how the structure of the xylem helps and function. We talk about that. Usually the xylem structures have a very large vessels with a very thick cell walls, so to speak. Why? Very simple, very high pressure. Because think about the regular, I have oak tree right there. So oak tree about what, 25, 30, maybe even more feet tall. So the roots have to push the water from the ground all the way to 40, 50, 60 feet high against the gravitational pull. That's create a lot of pressure. A lot of pressure. The vessels have to be relatively large diameter and also very reinforced. And this is what we see in the xylem. You will need to draw the side root of the under the microscope. So essentially, I think we have the picture. So here we go. And in this particular case, what you will see is the dermal tissue, the layer outside. That particular kind of triangle thingy is nothing else but the conductive tissue. The big vessels, the pink ones, let me increase. The pink ones right here, that's the xylem. The phloem will be right next to it. Everything else is dermal tissue. So you can point it out saying that's a vascular bundle. The outer part is the dermal tissue. Everything else is the something which is known as a cortex. And this is what it talks about, the same thing when you're looking at the, so you can see vascular bundle, epidermis, so dermal tissue, and everything between the dermal tissue and vascular bundle known as the cortex. All right, so root can be classified. Oh, and this is the microphotograph of the root. You can see the zone of division, zone of elongation, essentially, and whenever you start seeing the root hairs coming out, you'll see a zone of maturation. Again, root hairs are necessary to do what? To drastically increase surface area, to increase the absorption rates. More absorption, more water, more food. Now here we're looking at the different types of the roots. The taproot, the main difference between two of those. Look, the taproot, you can see the primary root with the secondary root coming out. On this case, in the fibrous roots, they all kind of fibrous looking one. You cannot see, you cannot determine which one is the actual taproot. And then there is adventurous ones where you can see that the stem goes along the ground and then the roots are coming in as well. So here we have adventurous root, and I think there is a, yeah, we're looking at the taproot, fibrous roots, yes. And here we have some of the pictures, so clearly can see the taproot, right? Clearly see the fibrous ones and clearly see the adventurous coming out. So here we have adventurous. Yeah. And then the nodules. Now, some of the plants creating something which known as the symbiotic relationship with the bacteria. Okay. Bacteria literally lives inside of the roots. They're creating that particular bulbous round structures known as the root nodules. And if you cross-sect, if you cross and look under the microscope, this is the root. This is the, of course, the large vesicles. That's a vascular bundle. So that's a xylem, phloem in between, the cortex, the epidermis. And now we're looking at that structure. That's a nodule. Within the nodule, we'll see a cells. Within the cells or right next to the cells, you will see the bacteria. Those pink ones are not the actual bacteria. The bacteria are much, much smaller. You don't really see them. And there one of the questions will say, does the bacteria gram-negative or gram-positive right here? You can say, well, it's some of the students saying, well, it's purple, so it have to be a gram-positive. We're not looking at the bacteria right here. It's kind of misleading, the slide itself. Bacteria is extremely small. We don't really see them under this magnification. So just believe me, just take my word for it. I didn't lie so far as I know to you guys. The bacteria is gram-negative. It's a rhizobium bacteria, which will be found inside of the roots, and it's a gram-negative. So what exactly we can see? You cannot see the rhizobium bacteria. You can see the nodule, the whole that particular thing. You can see the root itself. Bacteria helps the plant. How? We kind of talked about that a little before, but kind of giving you the idea. Bacteria lives inside of the root, so it's getting the shelter and the food. The plant feeds it sugar, which is producing through the photosynthesis. Bacteria is happy. It's a nice warm environment. It's pretty nice, lots of food, and they are protected. Now, what the plant is getting out of it, those particular type of the bacteria, the rhizobium bacteria, are able to take a specific compounds of nitrogen and convert it into the nitrogen-containing compounds so the plant can absorb. And then, of course, plant will start taking those particular compounds and creating its amino acids, and amino acids moved into the proteins and so on and so forth. So bacteria gives the nitrogen containing compounds absorbable or being digestible by plants, so to speak, and plants gives the place and food for the bacteria. So it's a symbiotic, more than that, mutualistic relationship. Quid pro quo. You give me that, tet for tat. Give me the nitrates, NO3, and I'll give you some food. No nitrates, no food. Make sense? So what type of the symbiosis? Mutualism, mutually beneficial. What does plant get from the bacteria? Nitrates, NO3, nitrates. What does shelter? See, sugars, glucose, C6H12O6. This is what the bacteria gets. How can tell you the rhizobium? You can say the professor told us so. No, technically it will be pinkish under the gram staining. It's not gram staining technique. That's why the plant cells are purplish, but it's not a gram staining technique. If you extract the bacteria, go through the gram staining technique, they will be pinkish color. All right. Any questions, any problems on that? So yeah, here we go. So this is the root, and if you're looking through the cross section of that, so here we're looking at the root itself, and then the place where the bacteria resides. Billions upon billions of those bacteria. Now, next table talking about the plant structures, and then we need to go through the epidermis, ground tissue, and then of course the vascular bundles. Vascular bundles, you see those particular kind of monkey faces. So the large openings would be the xylem. Again, thick, high opening, large diameter tubes to bring stuff up. The phloem, what about the orange one? That's the cells which will create the skeleton, so sclerenchyma. They'll support the vascular bundles. So the orange cells are sclerenchyma. So we're looking at the vascular bundle. The orange part is the sclerenchyma. Now the outer part would be epidermis, right? And then everything in between known as the ground tissue. So epidermis, everything between the epidermis and the vascular bundles are the ground tissue. So epidermis, the outer part, ground tissue, everything in between, sclerenchyma, the orange thing, vascular bundle, the blue and the red vessels. Phloem would be the blue, and the xylem will be large openings, and that would be the part of the vascular bundle, of course. Now, let's take a look. Which tissue, xylem or phloem, is closer to the epidermis? Well, epidermis right here. Let's take a look at this vascular bundle. The phloem is facing outwards. The xylem faces inward. Same thing. If you look at this particular vascular bundle, the phloem facing outwards, the xylem facing inwards. So the one which is closer would be the phloem. What is the function of sclerenchyma around the vascular bundle? Any kind of sclerenchyma will provide mechanical support. So that's a mechanical support of the vascular bundle. Make sense? Yes, all good. Moving on. Okay. Phloem, xylem. Make sure you understand that this one is showing, well, the xylem is this round red cells, dead cells now. It doesn't pointing right here. Some of the students I had before are like, why are you pointing? It's like, well, on the picture, it's pointing right here to this cell, so it had to be a xylem. No. It's pointing to that red, very thick tubes, essentially. They no longer living cells no more. So now let's take a look at the microphotograph. See, that's a cross section through the corn stem, and you're looking those particular kind of alien or monkey faces. So again, that one, the large ones right here, let's take a look. That would be a xylem vessels. The phloem will be right here, surrounded by sclerenchyma. Dead cells, dead cells, dead cells. Phloem is a living cell, sclerenchyma is already dead cells. In everything in between, the vascular bundle and the vascular bundle would be what? A ground tissue. Let's kind of take a closer look. Let me go back. See, in this particular case, the vascular bundles scattered throughout the whole stem. Monocot or dicot, that's a monocot stem. They are not arranged in any particular ring formation on the outer part. You can see it throughout the stem. Make sense? So yeah, make sure you will be able to identify whenever you're looking at the monocot or dicot. If you'll see something like that, the vascular bundles are not arranged in any kind of ring formation. That's a monocot stem. Now the dicot stem. Now, in the dicot stem, we'll have the arrangement. Those vascular bundles will be arranged in a very specific formation. It's a ring formation. And once again, we'll have the phloem on the outside, the xylem on the inside. The yellow part in between is the meristem, a lateral meristem. Remember, we were talking about the apical meristems increasing the length, while the lateral meristems increasing the width. So in this particular case, lateral meristem is basically fast dividing cells, If they ended up on an inner circle, so to speak, they will specialize into a xylem, again, large diameter vessels. If they will ended up on the outside, they'll specialize in the phloem. They are also supported by the sclerenchyma. So we have a vascular cambium, xylem, blue phloem, sclerenchyma. Dermal tissue. Now between the dermal tissue and the vascular bundle, that would be a thin layer of cortex, but inside part known as the pith. So let's take a look. Epidermis covered, right, on the outer part. Then the ground tissue, also known as the cortex. Everything between the epidermis and the vascular bundle on the outer side. Pith, everything inside of the vascular bundle ring. Vascular cambium on meristem, the yellow line, that's a fast dividing cell specializing into a phloem and a xylem. Vascular bundle, the whole that particular thing. It's a bundle of vascular tissue of the xylem, which will face inside, and the phloem, which will face inside, supported by the sclerenchyma. That's essentially what will turn into veins in the leaves later on. So here we go, the micro photograph of the helianthus, so the stem of the sunflower. You can clearly see the sclerenchyma, kind of reddish color. Then the bluish color will be a phloem. There will be a vascular bundle right here and the xylem. The middle part, the bluish part, is the pith. And of course, you can see the epidermis and then the cortex. Now, if you'll see something like that, you can see them arranged in a very specific, the vascular bundles, of course, arranged in a very specific ring format. That's a dicot. And if you magnify it, you can clearly see that's the part of the sclerenchyma. Here we're looking at the phloem. That kind of line which goes throughout the stem is the cambium. And we're looking at the xylem again, large diameter vessels. So epidermis, sclerenchyma, cortex, pith, vascular bundle, vascular cambium, xylem, and phloem. Already did that. All right. How are the vascular bundles arranged differently from the dicot to the monocot? In the dicot, they are arranged in a nice circle. In the monocot, they spread out throughout the stem. How are the vascular bundles similar? Well, they also contain the phloem and the xylem. Phloem is facing outwards, xylem facing inwards, and it's also reinforced by the sclerenchyma. Which tissue transport the water in the stem? Well, technically both, but water with the minerals is the xylem. What is the function of the sclerenchyma? Same as in the monocot stem, to reinforce the vascular bundle. Woody stems. So on the woody stem, you can clearly see the darker part of the wood, which is known as a hardwood, and the lighter part of the wood known as the sapwood. You can see darker one right here, middle wood, hardwood, and then the outer part is the sapwood. So here we're looking at the hardwood. You can clearly see the bark, and the cambium will be the line kind of right here. Yes, it's a living cell. So this is a dead cells. This is a dead cells as well. Only that part is essentially live cells, and the bark is the dead cells as well. All right, so again, we're looking at the xylem, phloem, which is the live cells, and then we're looking at the cambium, and the bark would be already dead. So dead, dead, alive. Very thin layer of the actual tree is alive. Well, we're talking about the tree trunk, I should say. So what's the difference between the sapwood and the hardwood? Hardwood already been blocked, all of those particular large vessels of the xylem being blocked off. As a result, it's used as the mechanical support. It's not conducting no more, even though it's a part of the vascular system, it's being completely clogged up. The sapwood is the one which is transporting water upwards. So here you can see the rings, the yearly rings. You can see 1, 2, 3, 4, 5, 6, maybe 7, 8, 9, and 10. So 10-year stem, 9 or 10-year stem. I don't know. This one is kind of double. No, it's actually separate one. So yes, it's a 10-year stem. And this is how it looks in a kind of cross section. So you can see the sapwood and the hardwood. Make sure to be able to identify those. Now the twigs. We're looking at the bamboo shoot, dried bamboo shoot. So it will be lacking the terminal bud. The terminal bud will be at the very tip of that shoot. In this particular case, it has been cut off. Okay, but what we can see on that particular part, we can see the auxiliary buds, the sideways, those ones, whoops, right here. That's the auxiliary buds. The leaves will be coming out. See that particular line right here? That's the node. This is another node. This is where, that's a third node right here. This is where the leaves will be attached. So this is the node, this is the node, this is the node, auxiliary bud. The distance between the nodes, known as the internode. So node, node, another node, another node. Auxiliary bud, internode, between the nodes. This part of the stem between the nodes is the internode. That part of the stem between the nodes is the internode. All right? And this is a twig, yeah, terminal bud. Just in this particular case, on the bamboo shoot, it was cut off. But we can clearly see the internodes, we can see the auxiliary buds, and we can see the attachment. Well, the leaves will be attached right here, so that would be the nodes. Now, speaking of leaves, type of the leaves, here we're looking at the simple leaf. One petiole, one blade. It could be a nice and round, it could be jagged, it doesn't really matter. As long as it's a one petiole and one blade, that's a simple leaf. In the simple leaf, you need to figure it out what kind of venation it has. Is it a pinnate venation, or is it all of those are pinnate venation, or is it a palmate venation? So see, here we have one petiole, one leaf blade, and a palmate venation. One petiole, one blade, and now we're looking at the pinnate venation because you have central vein and the secondary veins coming out on either side. Right? Okay? Any questions, any problems on that? Now, here we're looking at the compound leaf because we have one petiole and multiple blades coming out, or leaflets. The rachis is the place, is kind of a petiole between the leaflets. Now, in this case, it is the compound leaf. If you'll see the compound leaf, you will need to tell me the arrangement of the leaflets. Compound and the leaves going on either side. So it would be compound, pinnate compound. Everybody see pinnate compound? Yes. If you have a question right here, please make sure you understand that there is a venation and the arrangement of the leaflets which are using the same term. In this case, if I'll say what kind of venation it has, then you're telling me that's the pinnate venation. If I'll say what kind of leaf is that? Is it a simple or compound leaf? Be specific. So it's a simple, you're just going with the venation. If it's a compound, you're telling me the arrangement of the leaflets. And the arrangement in this case would be pinnate, and the venation would be pinnate as well. In this case, anybody knows what it is? What kind of plant, or what kind of plant this leaf came from? Most likely seen that. That's a cannabis plant, yes. So in this particular case, you see the petiole coming in and the leaflets coming out. But they're coming out as the palm of your hand. So it's a compound palmate leaf. Not venation. I'm not talking about venation. I'm talking about the leaf. Compound and then the arrangement. compound, and then the arrangement. Compound pinnate, compound palmate. That one, anybody knows what's this? Leaves of 3, let it be. That's a poison ivy. So again, we're looking at the petiole and then 3 leaflets coming out. They're coming out kind of like a palm, right? So palmate. Don't worry about this one because I had students before saying, well, maybe it's like if there will be another 3, it would look like pinnate. I would not ask about this, but make sure that you understand that it's a compound one, right? Everybody see that. So here we have pinnately compound. Pinnately compound because, well, it's a compound and the leaflet's coming out as the pinnate. And in this case, I actually put the sticky note in the face-to-face class. Students were getting it wrong for one or another reason. And I know the reason. They were matching up the veins. I was saying, is it compound or simple? They're like, well, that's the compound. I'm like, okay. And what kind of compound? And they're like, pinnate. I'm like, why it's a pinnate? And they're like, look at the veins. I'm like, no. I'm looking at the leaflets right now. So leaflets are spreading out for sure like a palm of the hand. Therefore, it's a palmately compound. Palmately compound with the pinnate venations, but don't worry about that. I usually don't mix and match. It's becoming too confusing for some of the students. So if it's a compound, you need to tell me if it's a pinnately compound or palmately compound by looking at the arrangement of the leaflets. Hopefully I got that down for you guys, hopefully. So again, we're looking at the coming in, the leaf, well, the petiole coming in, leaflet separates in the palm arrangement, therefore palmately. And then if I'll ask venation, if I'll be tricky, but I won't. You can see that each of the leaflets has a pinnate venation. And yes, if you want to be like, you know, kind of step up on your answer, says, well, that's a compound leaf with a palmate arrangement of the leaflets and pinnate venation. And I'll be okay with it, as long as you are doing it in the correct order. So again, here we're looking at the pinnate and the pinnate, right? So compound pinnate arrangement of the leaflets and pinnate arrangement of the veins. So don't mix and match. Arrangement of the leaflets, arrangement of the veins. Same terms. So a pinnately compound, pinnately venation. Palmately compound, pinnated venation. Now this is the parallel venation. So that's most likely we're looking at the, in fact, this is rice. Rice is a monocot. This is a leaf of the corn, also a monocot. So you can see that the veins are running parallel to each other. So here we go, the third type of the venation. So pinnate venation, palmate venation, and the parallel venation. So here we're looking at the pinnately venation, right? That's a tobacco leaf. Again, more of the pinnate. And now we're looking at the palmate. The petiole comes in and then spreads into a different major veins right here, again, like a palm. Now, this guy right here, a quick question for you. Well, first of all, we're definitely looking at the palmate venation, right? Everybody see it. Comes in, splits up into several major ones. So this guy, a big leaf, simple or compound? Look at this one and tell me, is it a simple or is it compound? Tell me what you think. Again, if you can do it in everybody's chat or in the private message to me. Simple or compound? We understand that it's a palmate venation in either or, but this particular leaf, is it simple or is it a compound leaf? Okay, only Jessica paying attention. Oh yeah, everybody else. Well, Astrid paying attention as well. And you guys are correct. It is a simple, even though once again, the blade is very jagged in this particular case, but that one do not reach all the way to the end of the petiole. If it would be reaching all the way, so it will be a simple, well, it will be a separate leaflets, that would be a compound. But in this case, it's kind of a simple right here, and then it starts separates, but it's still a simple leaf. So it is a simple leaf. Okay, please do not get distracted by those particular deep cuts inside of the leaf. It's still a one petiole, one large and fairly jagged cut out blade, but it's still one blade. So it is a simple leaf with a palmate venation. All right, cross section models. Now, in this particular case, you should be able to identify monocot versus dicot, and here you're looking at the dicot leaf. We already discussed that. The upper epidermis, the lower epidermis, palisade parenchyma, sometimes referred to as a mesenchyma. Meso means in between. In between what? In between the upper and lower epidermis. Palisade, the cells are located right next to each other, and then we're talking about spongy parenchyma. Now, whenever you're looking at the vein, this is what you're looking at. You're looking at the vascular bundles, which is reinforced by the sclerenchyma. So sclerenchyma, xylem, cambium, phloem. Let me kind of increase the magnification, and yeah, so let's kind of take a look on this. So upper epidermis, lower epidermis, vascular bundle, essentially vein, and the vein will be made out of the mechanical support, which is a sclerenchyma. Now we're looking at the large diameter vessels, that's the xylem, the phloem, and now we're looking at the yellow line is the vascular cambium, which basically produces vascular tissues. On that particular, on the leaf blade, we see the cells are located right next to each other. Palisade parenchyma, sometimes referred to as a mesenchyma, it's the same thing. And then the area where you have those air pockets, so under the microscope it looks spongy, hence the name spongy parenchyma or mesenchyma, same thing. Already work on that. So a simple draw a simple leaf petiole, leaf blade, mid vein. Okay, pinnately compound, palmately compound. Use the samples from the guide. Parallel venation, pinnately venation, palmate venations. Already did all that, and now we're going into a leaf. So yes, spongy, mesophyll, mesenchyma, or parenchyma, same thing. Don't have to worry about the terms. Don't the terms confuse you. So upper epidermis, the cells. That yellow line on top is a waxy cuticle. Then we're looking at the palisade parenchyma, spongy parenchyma, lower epidermis, and on the lower epidermis we'll have a guard cells. So let's see. Describe the orientation of the xylem in vascular bundles. Is it facing the upper or lower epidermis? Look, this is the xylem, this is the phloem. So xylem facing upper epidermis, phloem facing lower epidermis. What is the function of cuticle? Cuticle prevents the leaf from losing too much water, from dehydration or desiccation. What is the function of the mesophyll? Mesophyll is the stuff between the upper epidermis and lower epidermis. So everything, conducting, photosynthesis, storage, all of those particular things. Does the leaf has a vascular tissue? Yes, it has a vascular tissue, the xylem and the phloem, and it will be located where? In the vein. Vein is actually a vascular bundle. Where would you find most stomata? Stomata are the openings, and in this particular case, let me show you. So here we're looking at the different model. So that one, the very first layer is the cuticle. Then we have the upper epidermis, lower epidermis, palisade parenchyma, spongy parenchyma, and that one, two cells which can open and close because of the water pump in and pump out. That's the stomata. For the dicot leaves, most of the stomata will be on the lower epidermis. Question is why? Because the first part, this is where most of the sun will be absorbed, including heat. If it will be on top, it will evaporate a lot of water. Now, whenever you're looking at the monocot leaf, what you will notice that it does not have a palisade parenchyma. It has veins, and veins is made out of exactly the same thing, the xylem and then the phloem, and it's surrounded by the sclerenchyma. It has a mesophyll, also known as the parenchyma, but now it's all spongy. There is no cells which are a blade of grass. Think about the blade of grass, the cross section. So in this particular case, a blade of grass does not have palisade parenchyma. Why not? Because blade of grass essentially will be shined by the sun on either side. Whenever on a leaf, usually leaves have facing one particular side facing to the sun. So in this particular case, you're looking at the monocot versus dicot leaves. Presence of the palisade parenchyma most of the time indicate dicot. So I'll label the epidermis. Oh, and the stomata would be on either side of the leaf in the monocot. Mesophyll is everything between the upper and lower epidermis. So here we have the mesophyll, kind of light gray, light green, and here we have a darker green mesophyll. So here we have a bottom, and this is the stomata. So as you can see, there is a lot of them. So mesophyll, phloem, xylem, vascular bundle overall, sclerenchyma, of course. And remember parallel veins. Whenever they're looking parallel on the leaf, whenever you're cross-secting, you start seeing a bunch of the vascular bundles under the field of view, especially when you're looking under the microscope. And this is how it's relatively easy to distinguish between the monocot and dicot leaves whenever you're looking at the actual samples. See, right here, look, we're looking at the cross-section of the monocot leaf. As a result, here is the vascular bundle, here's the vascular bundle, this is a vascular bundle, this is a vascular bundle. Lots of vascular bundles. Because they are parallel, whenever you're cross-secting, you'll see more than one, or well, a dozen of them, essentially dozens, 1, 2, 3, 4, all of the vascular bundles, because the veins are running parallel to each other. Whenever you're cross-secting, you see larger veins, of course, but the smaller veins as well. That's the monocot right here. Now, whenever we're looking at the, again, monocot, see? The vascular bundle, vascular bundle, vascular bundle, vascular bundle, and so on and so forth. Vascular bundle, vascular bundle, vascular bundle, tons of them. That's the monocot leaf. In a dicot leaf, usually we see one or just a few of the main veins. So vascular bundle, and there is pretty much nothing there to the left or to the right. Not too many small ones. And here we're looking vascular bundle now, vascular bundle right there, vascular bundle right here. Those dark spots are considered to be vascular bundles, especially if it's a palmate leaf. You're cutting out, you can see several, but major, much larger vascular bundles. And also you can see that the first layer kind of arranged of the cells, arranged in as the palisade parenchyma, and then the other one has a much more empty spaces, spongy parenchyma. And this is what the side by side, the monocot, vascular bundle, vascular, vascular, vascular. That's a dicot. Vascular bundle, vascular bundle, vascular bundle. Monocot, vascular bundle, vascular bundle, vascular bundle, vascular bundle, vascular, 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 vascular. So you can see the difference between the monocot and the dicot stems, leaves. And here is a magnification, vascular bundle, monocot, vascular bundle, and bunch of smaller ones, a large one, smaller ones, larger one. This is a monocot. So di-mono. All right, what is the difference between the mesophyll and monocots and dicots? In the dicots, you have two layers of the mesophyll, palisade parenchyma and spongy parenchyma. In mesophyll, it's essentially all spongy. Do you see any similarities in the vascular bundles on a monocot and stem? Yes. In this particular case, how exactly the well, first of all, it's made out of both xylem and the phloem, and then kind of lower part of the leaf, the phloem facing lower part of the leaf, if it has a lower part. What is the main function of the leaves? Photosynthesis. Make sure you remember that particular reaction. Let me write it down for some of the students who still has troubles. It will bring you a lot of points, please, throughout the semester. Photosynthesis. Start with the carbon dioxide, right? CO2. Make sure you capitalize the first letter of the element. As a matter of fact, we'll start with the 6 of them. Plus, what do we need? We need 6 molecules of water, H2O. Plus, what else we need to photosynthesis? We have to have light. No light, no photo in the synthesis. Synthesis is not happening. Now we have the reaction going in the chloroplast, all that particular the electron transport chain, the electrons getting excited, leaving the reaction center, all that particular stuff, right? We're not going there, how exactly it's happening. Then it will produce something really nice and sweet, C6H12O6, glucose. That's a general formula of glucose. What else? Stuff which we are breathing with, O2. That's it. This is the reaction which I'm looking for. If you want me to overcomplicate it a little bit, here you can put 12 waters, and then there will be 6 waters at the end, but don't worry about that. This is the simplified version of the photosynthesis, the simplest one. What about cellular respiration? Same kind of idea, just in opposite direction. 6O2, or let me kind of go. I literally can copy and paste, right? This, and it will give me this. It's in the opposite direction. The only problem, the only issue here is that's not in the form of light. It's the energy in form of ATP. And that happens in the mitochondria. Again, the oxygen, well, the glucose molecule moves in, being broken down into pyruvates, right? Pyruvates moving further, start breaking down, hydrogen will be removed, carbon dioxide, you know, the citric acid cycle. We're not going there. As long as you will give me 2 of those reactions, I'm happy. I'm happy I'm giving you the full credit. That's all I'm looking for. They are opposite of each other. You know one, I didn't even type anything. I just copy and paste it just in the reverse order. And I substitute instead of light, which is a form of energy, into a different type of energy known as a chemical energy in form of adenosine triphosphate. Please make sure you know those. You will see them on every single or almost every single test, right? Can you give the examples of leaves that performs other function than what? Than photosynthesis. Thorns in the cacti. That would be the another function. Remember, we're talking about the flowers. They are modified leaves. For what? For sexual reproduction. Ta-da! And then you can come up with a few other ones. The storage. Think about onion, right? The onion which we're eating, that's the modified leaves for what? For storage, and for storage of what? Of sugars. That's why sometimes it's referred to as a sweet onion. And it's indeed sweet, especially when you fry to get rid of any kind of chemicals which make it kind of, you know, hot. So that's the storage. We are using we are using it in food because it stored some of the sugars. All right, anyway, observe the slide showing the sections of the dicot and monocot. We just did. How monocot leaf is different than a dicot leaf. Monocot has a bunch of smaller vascular bundles throughout the leaf, and usually monocot has dicots have a handful of the relatively large vascular bundles, aka veins. Post-lab review. What are the three tissues making up the plant body? The dermal, the vascular, and the ground. What are the three types of the cells which making up a vascular bundle? Xylem, phloem, sclerenchyma. What are the meristems? Fast-dividing cells. Essentially the embryonic cells. We have two of those: apical, which is increasing the length of the plant upward and downward. It could be sideways if it's on the ground, right? And then there is a lateral, which is increasing the width, girth of the plant. What is the function of stomata? Gas exchange. Carbon dioxide goes in for the cell, for the photosynthesis. Oxygen will be leaving the stomata into the atmosphere because it's a part of the, it's essentially waste product for the plants. Plants getting rid of that. How would you tell the difference between spongy and palisade parenchyma? In the palisade parenchyma, cells located right next to each other. Not much of the space between the cells, intercell space. In the spongy, it has a large air pockets to keep carbon dioxide and oxygen. Carbon dioxide will go into the cells, oxygen will go through the stomata out. What is the function of tissue found in the mid-vein? Mid-vein is nothing else but the vascular tissues, vascular bundle, transport. Xylem brings the water for the photosynthesis, the sugar from the photosynthesis leaving the leaves so it can be distributed throughout the body of the plant. In symbiotic relationship you observe, remember those rhizobium bacteria. What does the plant root gives to the bacteria? Shelter and food. What do the bacteria supply to the plant? Nitrates, nitrites, the nitrogen absorbable compounds for the plant. So the plants can later on modify it into the amino acids and then proteins. Do the phloem have nuclei? Yes, it's a living cell, so they do have a nuclei. The xylem, on the other hand, it's dead cell, so adult xylem is it's just a shell of the cell. Now, cell walls. Name two functions of the roots besides the water intake or uptake, the attachment to the substrate, so it will not be uprooted or anything like that. What else? We talk about the tendrils, right? Some of the climbing roots, so to speak. We talk about the air roots. We have here, the bold cypress. Bold cypress usually lives in a swampy area, so it creates the aerial roots. But also, we talk about something which is known as the storage roots, the sweet potatoes. Yummy. That's the plant storing its own food for the future use. We're coming in, biological bullies, digging it out and saying, no, we'll take that and we will eat it. You want it, but we'll take it from you. See? I'm simplifying, of course, we're not that mean. Oh, we are mean to the plants. So yes, here we go. Function, and you can look it up in the lecture as well. What is the function of the phloem cells? To conduct water with dissolved sugar from the part of the photosynthesis throughout the body of the plant. Now let's take a look. Complete the table below between the monocots and dicots. All right. So type of the root: tap root, TAP, tap root, or central root with a secondary root coming out. Here you have a fibrous root. you'll see a bunch of roots, and it's kind of hard to identify which one is the main root. Arrangement of the vascular bundles in a ring or around the stem, spread throughout the stem in the monocots. Leaf vein pattern, pinnate or palmate in the dicots, parallel in the monocots. Example of the dicot. Any kind of beans, okay? Beans, the peas, the snow peas. Then let's go into the peanuts, most of the fruits, not all, but most of the fruits, peaches, apricots, apples, pears, what else? Strawberries, raspberries, blueberries, and so on. Now, monocots. On the monocots, we are talking about many of the flowers. We see the lilies, we saw the tulips, right? Also, palm trees. Most of the palm trees are monocots. Now, I'm not talking about sago palm, but we're talking about like coconut. Well, coconut is actually, yeah, but in this particular case, lilies, tulips, irises, all of those are most of the grasses. Lots of grasses are monocots. Corn, that's the one we're eating. That's a monocot. Wheat is the, not weed. No, weed, the marijuana is the dicot, but wheat as the stuff we're making the flour from, that would be a monocot. All right, now number 12, your favorite. You doing actually, I looked through some of the labs, I think 7, 6, and 7. Looks actually fairly nice. You start getting what I'm asking you to do on the reflection questions. Please keep doing it. A nice short paragraph of what you learned, what you observed in this particular lab, and if you can apply or modify it in the future for your future for your future life. And if you can modify, if you'll think, well, it was really confusing, can you change that? I'm more open to any kind of suggestions. If needed, I will be doing it. All right, and we are done with the lab for today. As